Today we will be discussing a pure culture. This is a laboratory culture obtained of a single organism to test for disease. It is done with the streaking method. This consists of an agar solution, semi-solid, in a petri dish. You will need to obtain a solution from an inoculation in a medium that is mixed, that will go onto the edge of an agar plate. You will start out first by using a Bunsen burner with an inoculating loop hook. You will burn the tip to sterilize, sanitize, and let it cool. You will then obtain some of the specimen and put it on a quadrant of the agar plate, the petri dish. You will then go back and sanitize again, cool again, and go back, grab more of the specimen, go to the starting point of the bacteria, and spread and zigzag and space out the specimen to try to individually colonize this bacteria. We will do this over and over again until we've got the quadrants of the petri dish filled. Then we will do an incubation method. Once the petri dish is ready to go, it will be labeled, the lid on it, then it will be labeled, name, date, the person performing, its initials, name, what have you. And we will place that upside down in an incubator, and we will process this at 37 degrees Celsius for 24 to 48 hours to see what we come up with as far as the singled out bacteria, what we achieve. We need to know if it's growing or if it's inconclusive, meaning we had several different bacterias, we didn't achieve the desired results, so we would have to start over. If we receive a positive, that means that the colonies multiplied and growed and the production just is everywhere, and that would need to be treated. That is a positive specimen. If it is negative, that means it did not grow at all. It basically just stopped after incubation. So then, of course, based on those findings, doctors will need to know how to treat their patients accordingly, to see if they need an antibiotic, what type of bacteria it is by the shape, the movement, the size. Each antibiotic and treatment with certain bacterias go hand in hand. Some work better than others. And we need to know the length, the course, the time that it will take to treat what we're dealing with, what disease or infection that we have discovered doing the streaking method. And the doctors will then have to make that decision accordingly. They'll need to know how long they need to be taking the medication to clear up this, the findings that they did discover. And I do believe that is our conclusion. Thanks for listening.